heme portion is composed of Fe2 plus, that is a critical iron, and this globin is nothing but a protein. So now in that particular structure, this hemoglobin is basically composed of in between the four subunits of globin. So in this globin, there have the capacity to uptake of 4 hemoglobin in a unit. That means there are 4 O2 can be drawn in a particular hemoglobin. Now, in that particular experiment, we have to perform that how we can estimate that in a particular sample of blood, what about the percentage of hemoglobin in that sample? It is not possible in our way that we totally estimate the whole amount of hemoglobin that are available in our body. So for that purpose only, we are using some specific parameters that are used for determination of hemoglobin. So rather, if I am taking about the cell's hemoglobinometer, it is another name is acid hematine method. So why it is known as acid hematine method? Mostly in that experiment, we are taking blood from a sample, and in that sample we are using HCl for breakdown of RBCs that are available in that particular sample. That means we are using some some specific acid to break down of these RBCs. Why this acid is required in that case? Because of this acid will only reduce or breakage of that particular RBC surface. Now, my look upon concept on hemoglobin. This hemoglobin is only present into the surface and more specifically onto the globin chain. So if my permission is required to estimate about hemoglobin, then I have to remove this hemoglobin from the RBC. So it is the specific purpose for the breakdown of RBC1. And for that purpose, we are using acid. And this acid is nothing but HCl. HCl not in concentrated form. HCl not in concentrated HCl. Rather, we are using N by 10 HCl. That means 0.1 normal HCl. So this much of concentration is needed for breakdown of this RBC1. So for that purpose only, we are using HCl for breaking down of RBC1 and as well as extraction of the hemoglobin into the solution. So the acid hematine, that means whenever this hemoglobin is present, this hemoglobin is directly converted to acid hematine. How we can confirm that this hemoglobin is converted to acid hematine? This is nothing but the change of color. The change of color means initially it was in red, but now it is confirmed into the dark brown. By this means we can confirm that this hemoglobin is converted to acid hematine. So this acid hematine can be confirmed by the change of color of the blood. So if I am considering about several parts of this instrument, we have mostly different components. That purpose, we have a kit that is known as Sahli's hemoglobinometer. So in that particular kit, we have several components like one comparator box. In that comparator box, we have three chambers. The first and the last chambers are filled with some colored solution. And in this colored solution, you have to find they have more specific color like straw. Now, whenever I am putting the sample over here, I have to compare these three colors. So that's why it is known as comparative box. Next, for collection of the sample, we have one kind of pipette. This pipette is more specifically a marking on 20 microliter. We have to take this much of blood from the prick area, and this sample will be used for estimation. So whenever we are pouring this blood sample up to the 20 microliter mark, we have to compute that there is no air bubble. That is, no air bubble should be present in that particular synchronization. That means 20 microliter I have to take only blood. If there is any sample of air bubble, we have to discard the sample and fill it again. Now, these two, this one is the pipette and this one is the comparative box, as well as we also have hemoglobinometer tube. So what happened in that tube? We have generally two markings. Initially, the first marking is starting from 2, 4, 6, 8 up to 10. So, whenever this one is tube, hemoglobinometer tube. Why it is known as tube, not pipette? Because in case of pipette, its both ends are open. But whenever we are considering about tube, its one end is open and another end is closed. That's why it used as tube. Now we have to take this amount of HCl, N by 10 HCl. So we have to fill 2 G percentage HCl up to this mark. Now we have to take sample 20 microliter and then we pour the sample into this HCl. So now it will take up to the volume 2 marking. Now we have to stand over 10 minutes. For mixing it very gently. For mixing, we have more specifically one glass rod. So this glass rod is used for mixing of the sample as well as this blood and as well as this HCl2. After the 10 minutes, then we proceed about adding of distilled water. So what about the purpose of distilled water? Previously I told that in that case, we have one comparative box. In that comparative box, we have two different or the two colors are present two sides. The middle portion is open for inserting this tube. So now I have to check that how much amount of water I have to add to similarize these three colors. Initially, whenever I am not adding any kind of sample, this one is in red. Next, the dark brown. But after the conformation, after the adding of distilled water, its color will be changed to brown or in straw. So in that case, I have to add distilled water until which this three color will be the same. And now, whenever this value is getting, that means this three color will be the same. This is the final result of that particular experiment. And now we can estimate that this is the final value of this blood hemoglobin content. If it is coming like 6, then 6 Z percentage is the ultimate value. If it is coming like 8, then 8 is the ultimate value. So for the confirmation, I have to check one thing. The result is getting 6. It's okay, no issues. But I have to add one more drop in it. If the color fade, that means the previous reading was the same. If this color was not fade, in that case, I have to confirm that I have to add more water to equalify these three colors. So this is the basic principle that how we have to perform the Sahli's hemoglobinometer experiment. So hemoglobin is a very critical thing that we all require in our body for transportation of oxygen as well as CO2. Thank you.